So we've progressed from Mendel and his theoretical understanding of genetics, no idea what the genetic material was, to then understanding that chromosomes were the genetic material, but we still have a question in there. Chromosomes are made up of both DNA and protein. So this got to the question of what was the actual genetic material in terms of macromolecules. What in the chromosomes is the genetic material? And then once we figured that out, spoiler alert, it's DNA, right? That's why we have a whole chapter on DNA. Once we figured that out, then we got to figure out how it functions, which is a whole other set of problems. And we're still learning about that today. We don't have it all worked out. Even though we've been studying DNA for at least 70 years now, probably 80 years now, we still don't know everything about how it functions. So we're going to talk a little bit about the history of our understanding of DNA as the genetic material. If you want to learn more about this history, take my Micro 250 class. We talk a lot more about the experiments that go into that and things like that. We'll talk about DNA structure and sequencing. How do we read DNA? How is it laid out? We need to talk a lot about how DNA is replicated. Every time one of your cell divides, then we have to have DNA being replicated to give copies to the daughter cells. That's really an important process, and it has to be done properly. If it doesn't, if there are mistakes, then we get what are called mutations, changes in the DNA sequence. This also is the basis for the technique that we call PCR, polymerase chain reaction. So we'll look at DNA replication in prokaryotes first. That's where we best understood it early on. Then we'll talk about some of the differences when we look at DNA replication in eukaryotes. They're very similar, but there are a few differences there. Then I will tease that I'm going to talk about DNA repair, but this section does not make a lot of sense until we understand the implications of what happens when we change the DNA message. So we first will need to talk about transcription and translation, which is getting the information out of the DNA, before we talk about DNA repair. So I'm going to hold off on this one until a little bit later after we have a better understanding of those other processes. So what's DNA useful? Well, beyond the cool idea that you can understand the genetic code of an organism, I think that's pretty fascinating, right? You might think of DNA being useful in forensics or something like that, identifying who was where and whose blood this is, and somebody cut themselves while they were committing a crime, or while they, any of those things. That is totally a use, figuring that out. We have this new technology called CRISPR-Cas9 that you may have heard of. That is it's a mechanism to modify DNA. Now that's really powerful, right? Say you have sickle cell anemia. Let's use that. That's a basically single nucleotide that is changed that causes your hemoglobin to not fold properly. What if we could go in and cut out that nucleotide and replace it with the correct version? That could cure someone of sickle cell anemia. And there are trials of that going on right now, and it's going quite well, actually. What about history? Where did people come from? Where have they migrated to? What is their relation to one another? DNA can help us do this. There are whole companies that will try to tell you they can tell you about that. I'm a little bit dubious about some of it. We use our best judgment, but the science is in some places sketchy. We have some broad patterns that we see, but companies will try to sell you this service. I am not personally a fan of those things. We've seen companies like 23andMe come and have all kinds of problems, particularly with storing data securely. A lot of people's DNA got hacked. There's also questions of those genetics companies allowed police to search their databases, which allowed them to identify people through their relatives, which is kind of a scary dystopian thought. So that is an interesting ethical question. We have creation of things like vaccines. DNA, RNA have been critical in creating vaccines, and not just creating vaccines, but understanding how vaccines work, modifying them, all of these things. basic research. If you want to study anything in an organism these days, you're probably going to need to understand it at the DNA level, because DNA encodes all of the proteins that are in there and all the transcripts that are in there. So that is really important to understand DNA if you're going to do any sort of scientific research. Even if it's ecology, right? You're probably tracking different animals. You might want to know, is their genetic composition? Are they highly inbred, or are they outbreeding? How is the population health doing? How many individuals are in that population? DNA can allow you to do that. And then, of course, we have daytime television, where we have people like Maury Povich. I don't know if he's still on the air. He's probably really old, right? But figuring out paternity, and you got the people that go on the thing, and the baby mama says, I know he's the father, and he goes, no, I'm not the father, and then they take the DNA test, and you are the father, and he's all sad and disappointed, which is a strange commentary on our own world here as humans, our society, that we have television shows making a big spectacle out of paternity. But there we have it. You can figure out who the baby daddy is with DNA, right? So there are lots of uses. This is just my overview, right? So we will talk mainly about the basics of DNA. As you progress in your undergraduate career, you will probably learn more specialized uses for this. So let's back up. Let's go back to our timeline here. We're going to talk about a couple key experiments that lead us to understand that DNA is indeed the genetic material. So we've got to talk about transformation, which is a key process that allowed us to understand this. And then we will talk about Chargaff's rule, which allows Watson and Crick in some ways to make their model of the structure of DNA. So let's look at our timeline here. We have 1865. We have Mendel looking at his pea plants. And we're going to talk about this guy Miescher here in a moment. He's in 1869. Sutton and Bovieri, they proposed that chromosomes are the basis of heredity. So we got at least 40 years in there, right, where Mendel knows how it's working but doesn't know what's controlling it. And indeed, I don't believe Mendel was alive at this point. We will then go through this, and we will observe that there are a few key experiments in here, but we really don't get some good evidence that DNA is the genetic material until 40 years later. Here, Avery, MacLeod, and McCarty show that DNA is responsible for transformation here. So we then have Chargaff that notices this pattern here, and finally in 1953, Watson and Crick propose their model of the structure of DNA. So it's not until the 1990s that we even begin sequencing the human genome. So science takes a long time, right? And we still don't fully understand all of the information we got from sequencing the human genome. And we're still figuring out how it all works, even though we have the sequence. So let's start with the beginning. So I'm going to tell this story out of order because you know that DNA is the genetic material. I'm not going to try to pretend that we don't know that, okay? That's difficult. But there was a lot of question about that early on. So the first isolation of DNA as a molecule was done by a doctor/chemist, Friedrich Miescher. He isolated this molecule that he called nuclein from cell nuclei in the 1860s. So this is near when Mendel's doing his work. He doesn't understand the implication of this. It's just a molecule he can get out of nuclei. Side note, interesting side note, he was using, he was a doctor, so he had access to bandages that had lots of pus from wounds on it, and he was extracting it from those pussy white blood cells. So fun fact where DNA was first identified. Okay, so we know there's a chemical, it's deoxyribonucleic acid. He called it nuclein. What does it do? No clue. Okay, nobody's really going to understand that for quite a while, but we know it exists. So let's fast forward to the 1930s. And people are starting to think about genetic information and how it moves around. So we've seen the fly room, Morgan's work, Sutton and Bovieri proposing that chromosomes are the genetic material. Griffiths, he's a researcher, he's a microbiologist, he's studying these bacterial strains. So he has two strains of the same bacteria. They're the same species, but two different strains of it. One called the rough strain is not virulent. If you inject it into a mouse, nothing happens. The mouse lives. There's another strain of this bacteria called the virulent strain that when you, and it's the smooth strain because it looks smooth under a microscope, when you inject it into the mouse dies. Okay, interesting. We know that there are virulent bacteria that can kill mice, and by extension people. Not a super interesting experiment. They're different. The question that is interesting here is what makes them different. So, Griffith takes the smooth strain and he heats it up, destroying it, okay, killing it. The cells are literally exploded. He injects it into the mouse and the mouse lives. So he's wondering, has the information that made this smooth strain virulent been destroyed, or is it still there? So he does something that I don't think I would imagine doing, but he takes his non-virulent rough strain and he mixes it with some of the heat-killed smooth strain. Something strange happens here. He injects it into the mouse, and now the mouse dies. So it seems that he has somehow transformed the non-virulent strain into a virulent strain of bacteria. And he calls this property transformation because he changed it into a virulent strain. What's weird is Griffith has no way of explaining this. What is doing that? He has no idea. And unfortunately, he would not live to see this solved. So he never figures it out. He knows this can happen, but he doesn't know why. Again, you want to hear more about this story, Micro 250, we talk more about this in detail. We're going to jump ahead to a group, Avery, MacLeod, and McCarty, who are working, and they're fascinated by this transformation principle. They're doing this many years later, and they're going, okay, what could be the molecule that's causing this transformation to occur? So they looked in the nucleus, and in the nucleus, there are three types of molecules. We have DNA, we have protein, and we have RNA. Okay. The big question here was, is it DNA or protein? RNA is in there because it's a nucleic acid, but chromosomes made up of DNA and protein, but RNA is in there, so we got to eliminate that. So they predict that it is one of these three molecules, right? DNA, protein, or RNA is the genetic material. So what they do is they take the different strains, right? They take the smooth strains, the heat-killed smooth strains, and they go, okay, there must be the information in here. We're going to eliminate each type of molecule one at a time. So into the first tube, they add a proteinase. Proteinase is an enzyme that breaks down proteins, okay? So then they break down the proteins, they add back the R cells, transformation can still occur. That means that protein was not the genetic material. They destroyed all the protein, and transformation still occurs. How about RNA? Well, in a separate tube, they add ribonuclease, which destroys the RNA. They add the rough cells, transformation still occurs. Okay, it was not the RNA as the genetic material. In their third tube, DNA is destroyed by deoxyribonucleases. In this cell, they add the R cells and no transformation occurs. This to them is really good evidence that DNA is the genetic material. This actually wasn't enough to convince a large number of people. DNA just seemed too simple. Protein, it's got 20 different amino acids. That seems like a better way to make a language than 4 letters, right? A, T, G, and C. That seems strange. So this was not the be-all end-all. This is a great experiment, but there were a lot of complaints about it. Again, Micro 250. Talk more about it there. Okay, so I'm going to jump ahead and tell you what's going on here, right? Because you can assume, and you're willing to believe, that DNA is the genetic material because you've been told that, and we've done numerous experiments about that, and we know DNA is the genetic material. So what's actually occurring here? Well, the process of transformation is very interesting. Bacteria will occasionally just take DNA from their environment and bring it into their cells and add it into their genome. Now, that might seem weird. We don't like to do that because that puts us at risk of viruses, but this transformation principle can add new genes to bacteria. It gives them genetic diversity. Bacteria don't do sexual reproduction, so this is a way of introducing genetic diversity into bacterial populations. Okay, that's interesting. Is it really useful? Like, why am I spending so much time on this? Why am I so excited about transformation? Well, transformation, you can induce it. You can make bacteria take up DNA from their environment. So we can make them take up any gene we want. That could be really useful. Like, say, if you wanted to use bacteria to make a human protein, like insulin. The human insulin protein is critical in regulating your blood sugar. I think we're all aware of this. It is a protein hormone. Diabetics cannot produce the insulin protein. There's a couple of reasons why that doesn't happen. Some of them are genetic, some of them are induced through diet and things like that. There's various ways this can happen, but diabetics need to have supplemental insulin to regulate their blood sugar. Some of the earliest attempts to do this utilized insulin from pigs and cows that were killed and sent to the slaughterhouse. Okay, so you can extract the insulin from the pancreas of a pig or a cow in a slaughterhouse, and that insulin, the protein sequence, is close enough to be functional in the human body. Okay, cool, problem solved. Well, not really, right? If you think about it, every time a diabetic needs insulin, a pig or a cow has to die. That does not seem sustainable. That does not seem cheap. And there were cases where people were allergic to this because other molecules might come with it. So that's not ideal. What we would want is human insulin, real human insulin. In the 1980s, this is where transformation comes in. You can take the DNA sequence from the human genome for the insulin gene and cut it out. And you put it into a little circle of DNA called a plasmid. This process of transformation, we can cause bacteria to take that plasmid into their cell. We then take those bacteria and grow them in a big bioreactor, and they'll make the insulin protein. We extract that, and now we have 100% sequence identical human insulin. There is no difference in sequence, right? We took the gene straight from humans. This is amazing, right? You can take a gene from a human and a bacteria will make the exact same protein that a human would make. That's amazing, right? And now we have basically limitless supplies of insulin, theoretically. Drug companies still like to make money because really smart people had to think this up and make it happen. And then drug companies, executives want to make money and for shareholders and blah, 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 capitalism. So insulin is still probably not as cheap as it could be, but it's much cheaper than it was when we were harvesting it from pigs and cows. So progress, somewhat. You can make anything this way. Components for COVID vaccines are made this way. All kinds of stuff made this way. So this process of transformation, very important for just studying proteins in general, but also making proteins that we can use. Okay, back to my story about DNA. Okay, so like I said, people weren't convinced that DNA was the genetic material, even though they just did an experiment where when they degraded DNA, transformation occurred. And there were complaints about contamination, blah, blah, blah. Really, it boiled down to DNA has 4 letters, A, T, G, and C. That does not seem like an efficient way to store information. Remember, this is pre-digital computers, right? We think, oh, information, it's ones and zeros, right? That makes sense to us. You can store information that way. But people this time were thinking the written letter, right? Books. If you had a language that had 4 letters, your words would have to be really long, right? It's kind of like the Hawaiian alphabet, has a limited number of letters to it, so you get really long words like humu humu, nuku nuku, apua, right, for their state fish or whatever. That kind of thing is what people were thinking. They're like, proteins have 20 amino acids. That's more like a language, right? We have 26 letters. Why not 20 letters? Seems like that would be the genetic material. So, of course, we need experiments to settle this. And it came down to Alfred Hershey and Martha Chase, who you can see here. They worked with a system that we call bacteriophages. Okay, a bacteriophage is a virus that infects bacteria. Now, we've talked about this previously in this course. Viruses are not living. They're just a mix of protein and genetic material. That means that they have to infect other cells to replicate. Now, this is really nice, right? Because viruses are made up of just protein and DNA. The two things that we think are genetic material, okay? So we're going to use them to figure out which of these pieces is indeed the genetic material. So how do phages work? Well, here's a phage, and this is an E. coli cell, so you can tell this is an electron micrograph, right? This is really small. If this is the size of an E. coli cell, the virus lands and it injects its genetic material into the bacteria, causing it to create copies. What is the genetic material, though? Is it the protein or is it the DNA? I know you already know, but they didn't know. Okay, that's the big question. So how do you go about addressing this question? Well, in the 50s, what you needed to do was find a way that you could label DNA and label protein. This labeling comes in the form of adding a radioactive isotope to the molecules. Okay, DNA has a large amount of phosphorus in it, okay? The phosphodiester bonds that hold the bases together, we'll talk about, there's phosphorus in there. So what they did is they added radiolabeled P32. That's an isotope of phosphorus, and it caused all of the DNA to be radioactive. In another tube, they labeled sulfur. Sulfur is found in an amino acid, and that will label the protein. No sulfur in DNA. Okay, so we have ways of detecting these, and you can use photographic film to detect whether it glows or not. So what they did is they labeled P32 into some viruses and S35 into others, and they had two separate ones. Okay, they let this infect the cell, and they then waited for the genetic material to go in and waited for new phages to be made. They removed the old phage, okay, and looked. Some of the genetic material that goes in should remain inside of the cell. So they found radioactive phosphorus inside of the cell. In this case, though, they let the phage land and then they washed it off. The new phages and the inside of the cell did not contain any radiolabeled sulfur. The protein was not going inside the cell and therefore must not be the genetic material, while the DNA was going inside the cell and was the genetic material. So this is a radiolabeling experiment, not done as frequently anymore, but effectively a good way to track molecules. And so through this experiment, they were able to confirm that indeed, yes, it was DNA that was the genetic material. Okay, finally, right? At this point, people are willing to believe. It's weird though. How does DNA act as the genetic material? It doesn't seem logical. What's the structure of it? How is it containing this information? That's the big question. And some of the first people that started addressing this were not biologists, they were chemists, okay? Because this is a chemistry question. Chemists were doing all kinds of experiments with DNA. One of them was Erwin Chargaff. Here he is, very dashing man here in his suit and tie. Chargaff was doing experiments on DNA, and he noticed something interesting. When he got DNA from different sources, the amounts of the different bases that were in there were different. So like humans had a different ratio of A, T, G, and C than sea urchins. Back to sea urchins, right? They're still useful. He noted that, there was 29% A in the human genome and 32.8% in sea urchin DNA. 20.7% G and only 17.7% G in the sea urchin. Okay, that's something. What does it mean? Well, it didn't mean much until he noticed that regardless of where he got the DNA from, if you look in that species, the percentage of A is generally equal to the percentage of T. So let's look at corn, maize here. 26.8% A, he got about 27.2% T. That's 0.99. That's about as close as you can get to being equal in a real world. G, on the other hand, 22.8 to C, 23.2. That's real close to 1-to-1. And if we go down this table, they're almost identical, barring any sort of experimental error. So we call this Chargaff's rule. Chargaff's rule is the amount of A is equal to the amount of T, whereas this amount of G is equal to the amount of C. Again, an interesting finding, but it doesn't mean anything yet. But it's a clue to figuring out a piece of the puzzle, at least. So we will examine that in more detail in a moment, but let's do a little review here. So you go out and you discover a new species of tropical plant. And you're looking at the DNA in there, and you find that 20% of its genome is adenine. What percentage of its genome would be cytosine? Go ahead and pause the video, think about that. Okay, so we have 20% adenine. That means we have also 20% thymine. That's 40%. The remaining 60% must be G and C. So if those are equal, divide 60 by 2, and we get C, 30%. Why does this matter? Well, it's reinforcing something, right? Reinforcing Chargaff's rule to you. So to sum this up, DNA had been known to exist for a long time, since the time of Mendel. But it wasn't known to be the genetic material, so nobody was really studying it. Griffith, one of the first people to interrogate this question of what is the genetic material? How is it passed on? Observed transformation. And he found his virulent bacteria could transform the non-virulent bacteria when he mixed the strains. Avery, MacLeod, and McCarty used enzymes to degrade the different biological macromolecules, either protein, RNA, or DNA, and found that when DNA was degraded, no transformation could occur. Good evidence for DNA being the genetic material. Alfred Hershey, Martha Chase, they used bacteriophage with radiolabeled DNA or radiolabeled protein, and they were able to narrow down that DNA was the genetic material injected into the cells to cause them to replicate the phage. Meanwhile, Chargaff noticed that the amount of A in a DNA strand was equal to the amount of T, and the amount of G was equal to the amount of C. This sets the stage for the race to discover the structure of the DNA double helix, which we will talk about in the next section. All right, that's it for now.